Hi everyone, so this lab is going to be about electrophilic aromatic substitution using a bromine source, okay? So let's start with just the bare bones of what an EAS reaction is. So typically you would use a benzene derivative, so in this case we're just going to use regular old benzene. You'd react it with some halogen and a Lewis acid and you would form a halogenated carbocation intermediate and then you would reform that pi bond to reestablish the conjugation of the ring. So typically we can accomplish these kinds of transformations using a halogen source and some type of Lewis acid that has the halogen attached to it. So in this case, since we're using bromine, we have to use iron tribromide. Now we can also do this with chlorine, so chlorine and iron trichloride, but we can't actually do this with iodine. Iodine or fluorine for that matter, we have to do a different set of conditions to accomplish that. So we can actually dictate where our halogenation occurs depending on what kind of substitutions are already on the aromatic ring, okay? So I'm going to draw two examples here first. Two of them are an alcohol and an amine. Now, the thing to note about these two is that I'm drawing them having a hydrogen on them. I'll later fix it towards the end of the slide. But those hydrogens can actually be like R groups, so they can be alkyl groups, carbonyls, what have you. Now, these particular substituents are ortho-pair directing, so they tend to favor the halogenation at the ortho or pair direct positions. These are called activators. So I'll go on later on another slide and explain and actually do some resonance structures to help explain why these actually direct to these specific positions. Okay, so what I'm drawing now is what the typical meta director would be. Okay, so these actually favor the meta direction position. Sorry if you hear me clicking. Okay, so what does this mean? So meta directors aren't as strong as ortho-para directors. Okay, so they're slightly weaker, but they do tend to direct halogenation functionalization at the meta position. Okay, so it's really important to be able to identify what substituent is an activator and which ones are deactivators. Okay, so let's briefly do like just a reference, a generic mechanism, show you the arrow pushing of this. So we're going to go back to our basic benzene example, okay, from the first slide. So we're still going to do a bromination where we form this monohalogenated benzene, okay? So we use iron tribromide and diatomic bromine, okay? Now, one thing to note about this is that these two reagents actually form a type of complex, what I'm drawing right here. So this is actually the active reagent that does the electrophilic aromatic substitution, okay? So you actually have a positive charge on one of the bromines and a negative charge on the iron, because that iron doesn't like to have 4 of those bonds, or it's not neutral at that point. So we're going to have the electrons from that pi bond of the benzene attack the electrophilic, I should say the bromine that's just kind of dangling off there, and that's going to break that bromine bond and force the electrons from there up onto that bromine, okay? So we get to this carbocation intermediate where the bromine's attached to one carbon, and then you just have the carbocation at the ortho position. Now, you still have this hydrogen from the initial benzene left, okay? So how do you think you would to get from here to your product, you have to reestablish that conjugation, right? So how do you think you would do that? So you can actually use a base, right? But we don't actually add a base in this case, right? So we actually have that bromine that's from the that you push the electrons onto in the iron complex, and that'll come and abstract that hydrogen, and you'll form HBr as a byproduct, okay? So I don't actually draw that, but that's what typically happens, okay? So what if we used a hydroxy group as a directing substituent, right? So you'd actually form two products, okay? Now the ratio of these are kind of iffy, okay? They can vary from substrate to substrate. So you'd actually get the ortho substituent, okay? Sorry, I'm just messing with like the settings of the pen for drawing this, okay? So you have a halogenation at the ortho or para position for a hydroxy group, okay? So let's shift gears and talk about why those positions specifically, okay? So if we think about the hydroxy group on a benzene ring, okay, so that oxygen has lone pairs on it that can be donated into the pi system, okay? So if you donate a lone pair there, you end up forcing that pi bond electron pair up onto that ortho carbon, and that'll get you a negative charge at that carbon, okay? So in order to do electrophilic aromatic substitution, you have to have electrons at high electron density go to a position of low electron density, right? So in order to abstract that bromine, you have to have electrons somewhere, okay? And it has to be high density, okay? So you can also do arrow pushing and form another resonance structure where you have that negative charge at the para position, okay? So that's where you get your ortho para. You have electron density at both the ortho and para position, okay? So what about nitro groups? So you saw me draw just NO2, but in reality, it's what I'm drawing here as follows. So this picture is kind of easier to understand the movement of electrons. So in this case, nitrogen doesn't have that lone pair that oxygen does, right? So you have to. So in this case, you're taking electrons from that pi bond and and forming another bond with that nitrogen, and that'll force the electrons back up onto that second oxygen. Okay, but when you do this, not only do you pull electrons out of that that ring system, you're forming that carbocation on that ortho position. Okay. So that's the main difference between meta and ortho para directors. Okay, so if you notice in this particular resonance structure, you have a negative charge on the ortho position for the ortho para and a positive charge for the meta directing. Okay, so that means that there's less electrons there at that position to do the electrophilic aromatic substitution. So you're not actually going to, so if you just had like this nitro group, you're not going to see substitution at ortho para. So what about this particular experiment? Well, this experiment, we're going to be using vanillin as our starting material. So vanillin has 3 substituent groups on it. And I'll draw them here shortly. So we have an aldehyde group, we have a methoxy group, and we have an alcohol group. So based on this structure, there's 3 potential positions that you can actually do bromination. And in order to accomplish this bromination, rather than using bromine, so instead of using Br2, we're going to be using potassium bromate and HBr. Now, this is just a safer way to generate the bromine electrophile. It doesn't affect the mechanism at all. So in this case, you have 3 open slots that you can form your brominated products. And so you are going to generate three different products in varying ratios. Now, if we look at the substituents, each of these provide a different position that the bromine can be added, right? So we have an orthopara, we have 2 orthoparas and a meta director. So that will dictate where your majority of product is going to be. So things to note about this lab in particular is that you are using concentrated HBr, which is a strong acid. So you need to be cognizant of how you handle that. You also need to be aware of how much of your starting materials you're actually using. The one exception for this is actually the HBr in this case. So when you actually go to use the HBr, make sure to pull it up in the pipette and then squirt it back out into the bottle a couple of times. That'll help prevent it from dripping everywhere. So that should help mitigate any mess that you make, okay? Another thing is when you're going to like filter off your precipitate or in the lab, make sure to use the large receiving flask for that, for the filter, because if you use the smaller one, you just have too much liquid in this particular experiment for it to actually, to be um, um, necessary to use the small one, okay? So the small one just doesn't have enough volume for this particular lab. So safety-wise, things to note about safety for this particular lab is that you are using strong acids, so I'll write that up here. Another thing to note is that the potassium bromate that you're using in this lab is actually a fairly strong oxidant, so be careful when handling that. So general rule of thumb is that any like strong oxidant or reductant that you're using, it's always a safe bet to play it cautiously. So try not to spill it or make a mess of it when you're using it in your fume hood. And definitely, if you definitely don't avoid trying to spill it on yourself. Okay. So those are like the two main priorities that you have to keep in mind, is that you are using a strong acid HBr and you're using a strong oxidizer potassium bromate. So just be aware that those are the two main major hazards in this lab, and you should be okay.